Mutation and meiosis. Common exam questions and answers. Phenylketonuria is a disease caused by mutations of the gene coding for the enzyme PAH. The table shows part of the DNA base sequence coding for PAH. It also shows a mutation of this sequence which leads to the production of non-functional PAH. What is the maximum number of amino acids for which this base sequence could code for? Okay, so we know that a triplet, 3 bases codes for 1 amino acid. So that's 1, 2, 3, 4 amino acids, because there's 4 times 3, there's 4 triplets, so it's 4. Explain how this mutation leads to the formation of a non-functional PAH. There is a change in the sequence of amino acids. You can see here the G has been swapped for a C. This means there's a change, there could be a change in the hydrogen ionic disulfide bonds, which alters the tertiary structure or active site of the enzyme. Therefore, the substrate is not complementary, so no enzyme substrate complex is formed. Explain the importance of meiosis in the life cycles of organisms which reproduce sexually. Meiosis halves the chromosome number from diploid to haploid. When gametes fuse at fertilisation, the diploid number is restored. This keeps the chromosome number constant from one generation to the next. This introduces genetic variation because of independent assortment or crossing over. Figure 1 shows a life cycle of a moss plant. This is from the AQA past papers, these figures by the way. Figure 1 shows a life cycle of a moss plant. In this life cycle, only the stalk and spore capsule are diploid. All the cells in all the other stages of the life cycle of the moss are haploid. Which letter, A, B, C or D, in figure 1 shows where meiosis occurs in the life cycle of the moss? Write the appropriate letter in the box provided. So remember, it's where it goes from 2N to N, where it goes from diploid to haploid. And it says only the stalk and spore are diploid. So the stalk and the spore are diploid, the rest of them are haploid. So only the stalk and the spore capsule are diploid. So this one is the only one that is 2N, this bit here. So where it goes from 2N to N, because this bit here, the stalk and the spore capsule is 2N, everything else is haploid, so where it goes from 2N to N is the place where meiosis occurs, so it must be D, because it's going from a diploid here to haploid here. Figure 2 shows a cell from the moss plant. The cell is in the second meiotic division. So it's already separated the homologous chromosomes. What is the haploid number of chromosomes for this species of moss? Because it's in stage 2, it's already haploid. So it's 1, 2, 3, 4, 5, 6. Because although it's got chromatid, there's only 6 chromosomes. Yeah, so it's 6. Oh, I've missed one out. Explain how the chromosome number is halved during meiosis. Homologous chromosomes, the pair of homologous chromosomes, one of each pair goes to each daughter cell. That would happen in meiosis 1. Crossing over greatly increases genetic diversity in this species of moss. So, like, explain how it greatly increases the diversity. So, describe the process of crossing over and explain how it increases genetic diversity. So homologous pairs of chromosomes associate or form a bivalent. A chiasmata is formed. And equal lengths of chromatids or alleles are exchanged, which produces new combinations of alleles. So that could be a 4-mark question. Homologous pairs of chromosomes form a bivalent or chiasmata. Equal lengths of chromatids or alleles are exchanged, which produces new combinations of alleles. Describe and explain the processes that occur during meiosis that increase genetic variation. Homologous chromosomes pair up. There is independent segregation. The maternal and paternal chromosomes are reshuffled in any combination. Crossing over leads to exchange of parts of non-sister chromatids or alleles between chromosomes. Both create new combinations of alleles.